I think we have quorum. Good morning, good afternoon, everyone. So the agenda, well, first, as usual, we're recording this meeting and post this on YouTube. It's continued to be followed up by a lot of people asynchronously, so it's important. If anything you want us to edit, we'll do that. Just reach out before the end of the meeting. So in the last few weeks, we've been discussing the contour of a data analysis, a first paper that will come out of the study, and we agreed to focus our effort on a data release paper initially. And last week we made some great progress. I think we agreed this week to try to start assigning a task for the contour of that paper. So I shared over the weekend and iterations on the files that Lucas, Sarah, and others had done. And so that's what I was hoping we'd discuss today. Is there any other agenda that anyone wants to bring up? Or? Okay. So if you go to the meeting invite that you should see on your screen, there's a link here that bring you to a Google Doc, and underneath there's a list of tasks Excel sheet. So if you go on this link, you should arrive on this page, and this is the same link that we've been using ever since. I just spent a little more time polishing it, and the link at the top here is the same Excel sheet that Sarah put together, and it's available here. And so the intent is to collaboratively work on this Google Doc the same way as we put together the review, and then with transparency about with who is doing what in this sheet. And so in this way, the Excel sheet is acting both as a to-do list for everyone and authorship credit so that we know who did what. And so it's important that if you're doing something, I think you listed it there so that everybody knows you're working on it. Feel free to make edits to those files. It's completely open to everyone to make changes. But that's the intent. And so I thought, well, we could, and the third set of files is something that Alex suggested as well. So all of the figures are placeholder at the moment, but they are, if you go to the link slide, so you see if you go on the figure, it's not a picture, it's actually a Google slide. And so if you click on this and you click open source, it brings you to the PowerPoint that Lucas was putting together with the overall plan at the first and a list of figures. Right now they are mostly placeholders, but the intent is for us to sort of have this PowerPoint with the list of potential figures that we can use and embed them into the document. So this trio of Excel sheet plus Google Sheet plus Google Doc plus Google Slides, it has worked very well for the review, and it allows everyone to put comments and to work together on the pieces of the document. So that's what I think. This is something that also Alex suggested. So that's what I think we should do to put together this paper. Is there any questions on this organization?
Any chance you can put the link to it in the chat as well, please?
I'll do that just now. And then, yeah, for references, I need to set it up, but we've used PaperPile in the past. It has worked okay. And so I think we should continue unless people have a really strong opinion on it. And so PaperPile is a plugin embedded in Google Docs, so it allows us to sort of see everyone's references. Okay, so what I was hoping we do again is to go through the scaffold of the paper. I know we did this last time, but so to make sure we are aligned. It follows the traditional data release paper, I would say. So the abstract is the first, and then there's background rationale, The first figure is something that Alex alluded to as well, a statement of problems. And then I started to put placeholder text. This is completely a draft is not constrained at all. But I think the intent was just to clarify what each section is meant to cover. So the first, I mean, we're going to have to talk about the why this dataset exists. And so, we have an entire review, but I think it's helpful to have this explain at the onset of the data release manuscript. And then obviously the next question is, how does this relate to the review itself? So I think it's important to point the reader to that piece, which is very, very much longer than this will be. And then, so the statement of problems is here. The relationship with the review is front and center. And then we should have a paragraph that explain what is the data set intended to which gap is intended to fill. Last time we talked about the importance of, well, first the cross-context comparability and the multimodal aspects. And so this is at the beginning of the piece. And you'll see that what I'm proposing we do is, as we discussed last week, is use this multimodal as a thread throughout the paper. Any questions on this framing? Yes, Sarah?
Just one thing I want to say is I'm always confused with this term multimodal, because to me it sounds like, you study vision versus audition or olfaction as a biologist.
I see.
And so I think you have to be very clear and at least operationally define that you mean different recording techniques, but biologically I find it a bit odd. Yeah.
But so in some ways, yeah, it's really good you bring this up, because those two sections are actually talking to the two angles, multi-context, or multi-sensory or so maybe it's so important that yeah, it should go in the figure. What do you think?
I think it would be good to explain, yeah.
Explain the different axis, multi-modularity, so we'll say stimuli, a technique, a context, something like that.
Yeah.
That's a really good point. And so, yeah, so as we write it, maybe we should include that to clarify. Any other questions on the start of the manuscript? So the exercise, the reason I'm going through is what I think we could do is we go through the paper and then we go to the Excel sheet and see who wants to take the lead on different parts. We already did this with the team yesterday about the methods, and so you'll see how this could unfold. And the next section is going to have to be about the experimental design, because we talked about what the dataset fill, and so that's the most important section, how the experiment were constructed. And there are many ways we could explain, but we'll have to explain the behavior cohorts and the stimulus context. I'm sorry. And probably we need a paragraph about the technique that we use and the coverage. But that will be the background and rationale will be to cover those things. Anything else that is missing in your opinion? Yeah, that's important. I think it's pretty straightforward. And then probably the most important sections of this entire paper is the methods. And the reason is you all are going to do analysis on this dataset, and this is your unique opportunity to get all of the details that I needed to do this analysis. And so with the team yesterday, we went through it. It might be a lot of things that are missing, but and so it's organized by it follows the experiment from start to finish with big headers. So first you start with experimental animals. So it will explain, and it looks like a normal paper, but I think we probably should have had more details than it's typical for a publication. Surgery, implant, ISI for locating the visual cortex areas, behavior training, so how the mouse were uh, habituated to the stimuli, the details of the visual stimulus, the stimuli parameters, so all of the meta parameters of the stim. So obviously all the sections are just placeholder at the moment. Uh, really detailed description of the recording modalities. I'm calling out a figure. Where is this? Here. But would explain the experimental design and the different each modality. So figure 3 could be NeuroPixel 1 column, Mesoscope 1 column, SLAP2 1 column, and then the top will be will be organized in columns with rows that shows the CAD design of each technique, an exemplary assets of how the recording look, and then uh, anything we should have to explain how the units are extracted, and then and so on and so forth. So um, all of these methods should be paired with parallel figures. And so the recording modalities, we have severals, NeuroPixel here, 2-photon, SLAP2. Behavior tracking is a modality in many ways. So we have multiple cameras. So we'll need to explain how this was done. And then we also have um, brain coordinate alignment that is done for some experiment. Yes, sir.
Just a quick question for the neurophysiology using NeuroPixels. Are we also going to have LFP data? Because that's something that's not outlined in the current kind of brief section. It's all about spikes.
Yeah, we do. It comes with the NeuroPixel.
So maybe that should just be included in case.
We record the NeuroPixel in 2 band filters, and those are, I think we already shared some of this. So I think what would be helpful is if all of you could look, like, first, here is a great time to suggest things that should be in the methods, but also like offline. Had comments, like, because what we really we don't want to spend the next year explaining this experiment and times to the entire world. I think it's the right moment to sort of write down all of those really little details. And so the team is already committed at the Allen in the next 2 weeks to write down what we're doing on the rigs. And so you're going to see this section grow really quickly. And so it will be really helpful if you, as it's being filled, also you ask questions like, oh, I don't understand what this means. Can you hand me more details on this? Because this needs to be very, very detailed. Any comments on the data acquisition so far? Because I think the rest is data processing.
I've one question regarding the the stimuli setups. So during the SLAP2, we had these pilot runs, right, where we had included additional control blocks and so on. And then there's this standardized paradigm that helps to compare essentially between these different mismatch types or mismatch experiments. And I was wondering if these additional SLAP2 protocols, we somehow have them in there. Do we streamline it to just have these data that really share the same, exactly the same sessions?
our intent is to include all, like, so both the pilot and the newly acquired SLAP2 data in the data release paper. So yesterday, for instance, Severin, we were reviewing with the Severins and the rest of the EFIS team the NeuroPixel data that is, so we're already almost done with the NeuroPixel data, and we're just reviewing the entirety of the data to just make sure there are no issues or we understand the issues and so on and so forth. And I think what you realize is, as you know, neuroscience experiments are incredibly rich. There's a lot. Anytime you acquire an experiment, there are some things that are slightly different, and so there is so much information from each session that is helpful. So, for instance, that day one probe was not as good, and so you have a slightly lower yield, and we know why, right? And so we're probably gonna produce really rich tables for each modalities that explain, we did this already, but I think we probably need to do even more. So we have this session table that we review already that show all of the rows and that are shared across modality, but I think we should also have modality specific tables, and some of these might need, as you said, this experiment as this design with this parameters, and you'll be able to use it. But it's really important that we share extensive data table with experiments and not typical, something like that.
You have to say,
but yeah, we're planning to.
In my experience, students struggled a lot with these early pilot experiments and then being confused why the scripts wouldn't run later on, and then that the context changed. And another thing was actually also some of the transgenic combinations and backgrounds were not trivial. So if you're like a aficionado, you will know what that one line means. But a lot of people who have no exposure in these different sensors and combinations of animal mouse lines, they can't necessarily follow easily, I have to say.
So what are you calling for? So do you want more explanation of each mouse line? Is that what you're asking, or is there a specific? How can I convert your comment?
Maybe so we have a list of the transgenic background and which sensors have been used, but a lot of times it doesn't say why. So, for instance, I had some students that were confused whether, the let's say the GCAM signal was synaptic or cellular, or basically what the different sensors were used for, because they didn't have a connection of why they know where a certain sensor is expressed, for instance.
Yeah. I'm just trying to bring up the mouse table.
Yeah.
We had these table here, the mouse table here.
Exactly.
So
yeah.
This is not clear, is what you meant?
So I can tell you for the majority of the third year students that I tried this with, it was not clear to them. It was not clear. Also, PHP, EB, and whatever or not, and so it's kind of like I know what it means, but I realize it's super compressed information on the table, and so there might be a way to make it easier.
Transgenic information with descriptions of what is what and for what reason. That's right. This is also like, I mean, this is a generic problems in the method sections of all papers. It's just the higher the journals, the worse it gets.
Not always.
Okay, but yeah, I think we should just be generous with the method section. I agree with you. And so, in fact, actually, if you could ask your students, Sarah, to write down those feedback, like in their own words, that would be really helpful.
Yeah.
And as I said, for this piece, there's an authorship list, but I think anybody that contribute should be listed. In my mind, really important feedback about these things qualify for contributions. I mean, if you spend time reviewing and adding comments and saying like this, I really need this information to analyze, I think this is enough to be a co-author in my mind. And so we just have to be transparent about what is what. That's all.
Sounds good.
Any other question? I think this is really helpful. Okay, let's go a bit about data processing. This is a complex topic. So we have 3 or 4 modalities. I mean, yeah, so the same way each modality is going to come with its own pipeline. And so we'll need to explain how this was processed. In addition, I think we will link the code that was used on CodeOcean, so you should be able to read the code. In my mind, English is a poor descriptor of processing, and so I think I'd rather have people read the code, but we'll explain it here. That's right. Anything in? I mean, this probably is too short. We need to add a lot more. I didn't realize it was so short. Where's my comments button? Subsection. Any questions on processing? Because I think the processing content of the paper should be bring you to units. So there's a really nice parallel between the techniques. So NeuroPixel give you, extracted using a unit. Mesoscope give you somatic ROIs, so not necessarily dendrites, and then SLAP to give you, connections ROIs. And so these are very different units, but they have all their own segmentation pipeline. So we'll explain it this way. And similarly, the first figure here would highlight the data could show those units in, so you have a first column that shows let's say the NeuroPixel units, and then the characterizations of amplitude, SNR, and yield of units. So that will be the intent of the figure, just show for each modalities how units were extracted and the quality of those units, and how many are likely noise and which one are more likely signals. And so that figure will be very important to sort of get an intuition about, the quality of the signal. Any questions on data processing?
Just a very quick comment. So one good example for using jargon was the ROI term, because I had lots of discussions where students asked me, what is an ROI? Is it a neuron, a dendrite, a synapse, and then all different ROIs belonging to some biological entity? And so I think we just have to be careful that we don't have X data sets which all have the term ROI, but they mean 3 different things in 3 data sets, or maybe they can always be ROIs, because ultimately that's what you extract, but whether it's a synaptic signal or somatic signal or dendritic signal just has to be super clear.
Do you think we need some kind of glossary?
I think it would be useful, yeah.
Yeah, because it's actually uncommon in my experience for like data set that bridge modality so much, and so each one has its own language, and so yeah, maybe we want to clarify what we mean for the people that experience with units, what is an ROI, and vice versa. It was a really good feedback. Thank you. So that's the method section.
So on the method section as well, probably one of the main thing which can be there would be code which is frozen and which people can, because no matter how much language you're going to dump into the paper, it will never actually cover every single parameter. And I think that it is very important to have a perfect DOI. And I know we went, for example, right now, we were going through a paper, and sometimes there is an mismatch between what you find in their code and what you find, for example, in their text, and you find out that they changed some settings or things like this. So having a frozen version of the code, which was exactly what was done for that publication, I think it would be very important to have as a supplementary material for the methods.
Yeah. So let's say provide the frozen. So Stefan, this is really good feedback. Well, so the way we use CodeOcean right now is every process asset grow through a, it's using a Docker, and then inside there's the code in the container and the input output parameters. The input parameters that I use are saved. So Carter is mostly doing that now. And I think we agreed to release public capsules that contains both the data and as well as the capsule the data that the capsule code that was used to run those process asset. But there is a bit of a challenge here is some of his data sets, sometimes a new version of the pipeline is pushed or we fix something for certain assets. And so we'll have to maybe make a table. I don't know how complicated it will be, but just a table that shows for each asset which version of the pipeline was used or things like that when it's not exactly the same. Yeah.
Well, just letting you know, we encounter this problem all over, right? So that is one of the biggest things to, you look the methods, you can't actually reproduce things from the methods because words are not generally spent enough to do that, and journals would never agree to actually publish enough methods to fully reproduce something. You open the code and they don't match.
Yep. Yeah.
To be fully, I mean, we have tons of files, right, across those three modalities. I think a wise thing to do is before the data release, before this paper it comes out, is doing like a re-upload of all files with the same version to ensure compatibility, because we've iterated so much throughout all these files. I think it will be a little bit laborious, a little bit annoying, but I think it'll be worth it to make sure that everything is consistent.
Yeah. Regardless, I think Stefan's point of like providing clear links where you can find the code that was used for each asset is important.
Yep. Just to clarify, maybe I'm not sure I understand what you mean by processing. Do you mean the pre-processing of the raw data that ultimately leads to, for instance, let's say units and spike times and things like that you basically release? Or do you mean the one step of like the basic analysis that's been run then on already that pre-processed data? Because I'm wondering, do you expect people to go through the, let's say, image segmentation and RI identification from raw data using fixed codes? Or do you basically provide as the output of that as the data release, or both?
So we'll provide both.
Okay.
We'll provide the raw data, we'll provide the process data, we'll provide the code that brings the raw to the process data. All of those three will be through CodeOcean pipeline.
Okay, got it.
System.
Okay.
Yeah. And what I think we keep these thoughts because after the data record section that we are about to discuss, then we have the validation. And the validation is as we discussed last week, we'll have to share some first order results about, quantifications of receptor field and things like that. And that will be on some of you to sort of do that. And I think you should all keep an eye on, okay, where is the code that I use to generate those plots? Can I publish it with the paper in a similar way? Right. Any other, yeah, any other question? This is really good. Okay, so this is the methods. And as I said, in my mind, this is probably one of the most important section of the paper. The next one is the data records. And so this is a bit unusual for a scientific publication. It's specific to scientific data paper. And so where you would describe the data set in its entirety. So first, we start by describing how the process data is available, but we might also want to explain the raw data section. I think this is speaking to Sarah's point. And so this is just a placeholder saying which file format we will be using. And then there will be a data table section where we'll have essentially figures that shows that for each modality, the list of experiment, list of animals. We already started this work, so I will be using this as a starter. This link table. I think we should also probably have a more extensive table for each modality. So this will be mice, a cross modality, and maybe a table for each modality. So that will be quite extensive. And then Carter was going to take a stab at explaining how the files are organized through Dandy, and probably as well as on the raw files. So that would be the content of this section. So this is useful when you want to start doing analysis. You want to see where is everything. And maybe, I don't know, maybe the glossary belong there. Maybe not.
I think it could be useful there because that's where you really try to go through the tables to identify the features. Right. So for instance, one example was specifically for SLAP2 data that everyone extracted DMD1s and DMD2s, but then realized a month in that sometimes DMD1 is an apical dendrite or a basal dendrite or something, and that was quite confusing, for instance.
And what does DMD means?
I think that was the first thing, which is, I'm not sure whether everyone knows at this stage. Yeah.
This is the first thing you get when you join the L Institute. We have so many acronyms, but we get, at some point we forget. Okay. Again, Sarah or anyone, maybe we should start this as a separate document, list of acronyms and all the things that we're confused about, and then we'll define it. Okay, so this is the data tables. So it's an entire section about organizing the data release. And then this is the next section, the data validation. And this is the one that we probably spoke the most so far. And I propose we organize into 3 main themes. I think that we've already covered, but the first one will be unit extraction. So validation of unit extraction. The second one will be receptor field across modalities. And the last one, I think, is behavior. Four section, behavior across modalities. And the last one is stimulus evoke response. So we could choose one simple of this context, one of the contexts, and just highlight what we see. And then we've talked about doing more complex analysis, but I think the consensus was this could be into a follow-up paper. But I want to make sure that you're on board with these. Lucas, you're on board? Yeah. And so the intent of the analysis is the unit extraction is to highlight the quality of the data. So, I get an understanding of essentially signal versus noise for each modality. And so the figure 4 here would be, again, as I just said before, modality per modality. So NeuroPixels, Mesoscope, and SLAP, and then you'll see, and we have to agree on what we want there. But essentially, signal to noise, some sense of stability, and some quality control metric. And different modality might have plot that is specific. So for instance, for 2-photon, we have a little more chronic recordings, although we actually did 4 sessions for NeuroPixels. And so, yeah, we could mix and match here, but that would be the intent would be to sort of parallelize the description.
What about cell type classification for the electrophysiology, like waveform clustering, or so? Is that something that?
Cell type classification. How would you approach that?
I'm not sure whether it would be part of pre-processing to give putative labels.
Maybe that's a separate supplemental, or do you want in the main manuscript?
Yeah, I just don't know whether you would call it quality, part of quality control or not, basically.
Yeah, we have code to do that. Oh, I think I know where it could go into the usage notes. Because it's maybe it's going beyond just like you could provide, data, you could provide an example code to do cell types, studying cell types.
Okay.
But that's a new section. Cell types code here. Okay. And so what we need to do probably in the rest of the meeting is just this, because I'll go over the Excel sheet after that, but decide who wants to start that when the data is available. And then the next one is receptor field across modality. I think it's a fairly common stimulus that a lot of people have very specific expectations. And so the same way, modality are columns and then receptor field. I think it will be really nice, right, to see across modality how the receptor field are different or the same, and even across areas. So I think it's fairly straightforward figure. And you could include zebra analysis in there as well. So 1 row for normal and 1 row for zebra.
Yeah. Marcel prepared a little outline for that figure. Maybe you can briefly, I don't know, if you like, we can have a quick look to discuss the scope, but we can also go through everything and then focus on that. But one thing I was wondering, I think for these last figures, it would be nice, as Stefan was also saying last time, that it would be good to have a consistent layout where we go through the recording modalities. So maybe a column design would then be good, where we have, NeuroPixels and so on, and then we do all the things as rows, essentially then.
Yeah.
Maybe that's something
I mean, all of us figures are, that's why I sort of set it up on your Google Slides, Lucas, is you can sort of play on your own corner of the slides, and then as we get all of them, we can align them so that we have the same design.
Yeah, that's good.
Kareem.
Yeah, regarding the NeuroPixels recording, the stability across one session bullet, is that meant to ensure that the units are the same, or is that more to assess or analyze any changes in the information representation on a trial-by-trial basis? Is there 2 types of stability of signals and stability of information, right?
There are 2 types of stability. I think the intent here was the stability of the units, not of the signal, of the neuroscience signal.
Yeah.
Because I think we know there are sessions that have a little more drift than others. We already know. We have looked over this yesterday with the team. And so it's helpful for people to understand, like when you're looking at responses across the whole session, be aware that some units drift, some do not, and so it's an important things to have in mind. No
Right.
experiment is perfect.
Yeah, understood. So if that is the case, then we might want to think of adding another bullet down there to quantify if there is any changes in the information representation of the cells on a trial, because RFs basically are averages, right, across all many trials. And so,
so you were here, you would in the stimulus evoke response.
Yes, that's my point.
Because
I think when Nicholas did analyze these early on, he found some changes depending on the order of trials. So it might help.
Yeah, we should just be careful that if you start going in that direction, you're getting into really sort of deep biology and really interesting biology. And there's a a nice it's a trade-off. Where how far do we want to go for this paper?
Which relates to the question I asked you last week, which is, okay, this is a data release paper. So to what extent we want to, put more characterization about what's happening in the data or not. Yeah.
I think the first stimulus analysis figure is intended to show there is signal, there is interesting phenomena that we see, and in my mind, maybe showing across that there is some variability across context is as a first order maybe useful to sort of highlight, but I would not go too deep in the analysis, like starting to contrast it with the controls and then do sort of shuffle
No, that's not what I
analysis.
meant. It's basically if we're observing, the same set of units, let's say 50 neurons or whatever, and then we say, okay, here is this angle under the passive viewing versus the running, how the response properties do change in that regard. So just basically.
Yeah. That would make sense. Any other questions on Lucas? Do you have more comments on the receptive field? Or you're good.
No, I think I mean we would like to include the zebra because it's a cool feature of these sessions.
Yep.
And yeah, I guess we just post the proposal then on the Google slide so that you can check it and then we can discuss it. Yeah.
Yep. I mean, you could also like start putting the figures you have already in the Google slide. That's right, and then we can comment there and see what makes sense. The next section is behavior data.
I forgot one question. We were thinking whether it would make sense to extend that figure to also include basic tuning. So for example, because we have blocks that can characterize orientation tuning. I remember Sarah doing the polar plots for orientation tuning. One can do the same with the zebra. So in principle, if we say this is kind of let's say basic visual tuning or response properties, one could also add in principle example tuning plots and maybe add like spatial frequency tuning and size tuning or something that one can pull out of the zebra just to highlight that there's more more sophisticated features that we can extract about visual responsiveness.
I mean, maybe that belongs here, like in the columns, right? A bit of orientation tuning across modality. I think that would be helpful. That's right.
But do we have the zebra stimulus for all the recording modalities?
I think we do.
Okay, I wasn't sure.
The only one slap to in the end we switch a little later, but but it's the only. I think we do.
Okay.
I mean, we haven't analyzed this data in depth, but it's been recorded. The next is behavior data. I think we could highlight. So there are free cameras for all of the data set. And so some of this is going to be very rich. And so we could highlight the high tracking data and then maybe the running data and some of the behavior camera. and maybe just a very low order analysis of pupil across the different context. I think that could be insightful.
What about the motion energy of the face? Because
That's
that's
right.
been strongly associated with fluctuations.
It's just I don't know if we have the pipeline in place to do that. But if you want to take a stab at this, you'll be welcome.
Well, I guess it's recorded in the eye tracking videos anyway, right?
Well, we have a face camera.
Okay, so then anyway.
Yep. I think you're all gonna realize that there's so many things to look at, but we'll have to sort of make a choice. But yeah, there's likely insightful responses on the face to different types of stimuli that you could extract.
I just think in terms of the point of these figures, right, isn't it to highlight the different kind of ranges of parameters that are there, even if you're not going into detail, but just to say that there's information on these different behavioral metrics to some extent without having to go into a thorough analysis of how that specific metric correlates with any of the modalities.
Yeah. I'm not sure the mapping is so clear. Because in the behavior video, you can analyze, stress, you could extract attention, you could extract running state, there's very different types of state you could extract. And it's not always clear which one contribute to what in the neural activity.
Are you planning on uploading the face videos and stuff with the data release?
Yep.
Oh wow. Okay. Cool.
I just thought that in terms of data release, it would be useful for a person to know that, for instance, this data is part of this data set, right? And so if you have a visual that shows that there's information on encoder, pupil, and face, then it's more likely that people will pick it up and look into it than if it's just part of it and you don't know, right? You just focus on the physiological metrics from the EFIS or the imaging, basically.
Yeah, it's just we have some tools to extract, to segment the behavior videos, but I just don't know if they will be ready for this paper. That's the challenge, because it's a non-trivial amount of work to do this well. Yep, I'm just not sure. Check what is available for behavior processing.
I mean, worst case, just a visual, like let's say a screenshot of these different, like the videos or so. I mean, that's already like that on one glance you can see, okay, that's in there, and then probably every group has their own tool.
Yeah, the pupil, we have tools to track it and quantify it. The pupil and the running. It's just when you start about motion energy, I think there are tools that are available, but I don't know if they will work out of the box right away. Okay, I want to keep some time for the Excel sheet. So this is the content of essentially the manuscript. And then the last section is usage note, where we would provide some data access examples, so notebooks. We discuss limitations, and then we could have a little paragraph that explained the analysis plan or like future directions where this could go. Does this sounds right to you, or you have like major suggestions? Or okay, so this sheet here that is linked at the top links. So there is, I think it's trying to be current, and so this column here is the name of the section. And then yesterday with the team, we went through the methods, but is in light blue. And then I found internal people that will spend time in the next 2 weeks. We set ourselves a date to fill it in. And so you should expect all the methods to go in the next few weeks. What I thought will be helpful is to discuss these here, which is in green, so the data validation. And I see that people already put the names. That's right. Any questions there? I think anybody feel free to sort of express interest in contributing there. What I think would be great to happen is that those groups sort of self-organize when we share the data, and then you start putting together figures and plots that will go into the slides, and you copy paste here, and then we have a discussion from there. Does that make sense? Does anybody have questions on this sheet? Is something not clear, or?
I have a question.
Yeah.
Turn on my camera. Don't we already have plots on the signal quality for delta F over F?
I
Or
mean,
am I
we've
mistaken?
done. I mean, I think it depends. So you put your names here. So this is, or maybe I should clarify. This is mesoscope. Yeah. And this is slap 2. So delta F over F, we actually did, yeah, some plots for the SLAP 2 experiment with synaptic imaging. But we all very soon, probably in a week or so, you will have access to the raw data for mesoscope. And there we have ROI with, again, delta F over F extractions. And so we have to make quantification of its traces, and then and to get a feel about, the quality of the signal in those experiments. Does that make sense?
Yes.
But for SLAP 2, yeah, we have done some of these, but there will be more experiments to. I feel like ideally the plots are also including distributions across the entire set of experiment, right? So that you get a good sense of the quality of the units. Sarah?
One more question regarding the validation of the quality. Do we have to have motion correction metrics? So how is that handled? Is that already integrated in the pre-processing, and we just track how much correction had to be applied, or do we have to quantify how much motion there is, for instance?
So we have a motion correction algorithm that was run. You will have access to the output of it, and so you could create a plot. Motion for each modality will be different. So on NeuroPixel, we don't do motion corrections, except I think there's some unit drift. I think kilosort is a little more sophisticated now, but it's a different kind of motion than the one on the 2-photon that is more planar and a bit Z. Well, actually, the Z is the most annoying part, but we quantify these things, the X, Y, and the Z as well. And so we could decide to have a plot on motion across modalities. I think that's perfectly valid. I mean, there's a large space of things where we could explore. Any other questions?
Just a very small thing. For example, cell or row 39, the stimulus evoked responses, outpost standard versus deviant. Looks like you should rather move to figure 7. Is that, what do you think?
So just move the row?
Yeah, like it sounds more like that. Yeah.
Yeah, that's right. That's right.
and I guess that maybe I mean we can fine tune the.
I mean, if you feel like there should be a new row based on the document, please create the row. uh, I'm not going to think about everything. Please create the row. But I think the intent here is if you start to do some analysis, please create the row unless it exists already, and then make it apparent to everyone so that your contribution is clear. People can find where you put things, and that's what you wanted, guys, to have this Excel sheet that provide transparency.
Yeah, it's super. Yeah.
Yeah. And so I appreciate that you don't have access to the whole data quite yet. I think Carter is. Do you want to update everyone, Carter, on where we are?
Yeah. So for the EFIS, we're still waiting for QC. That's going to take, I think, probably multiple weeks, if I had to guess, before that gets uploaded. And the CCF is so the Common Coordinate Framework for the brain regions is going to take much longer. So we'll do an upload of the electrophysiology files without that first, and then we'll re-upload them later when we have access to the CCF. For Mesoscope, things are in a really good place. There's some small bugs with the packaging, but we're very close to having all of our Mesoscope stuff uploaded. So with good luck, it will be ready by this time next week. And for Slab 2, we have a lot more work on the packaging infrastructure. So that's probably several weeks away.
But Carter, do you want to comment on the raw data and the process, and just not only NWB?
Oh, yeah. So for EFIS and OFIS, they are on CodeOcean. So one of the next things we'll be working on is getting those put in the public collections on CodeOcean. And then we'll be able to release the raw data to you guys. It won't be packaged, but you'll have access to it.
Okay. So I feel like once we have the data access, we probably should go back to the sheet and help you guys get started, which is like 1 or 2 weeks away for some assets. So we're very close. But in the interim, we'll fill in the methods because this is what you need. And so it would be really helpful if all of you could go through the document and ask your questions. Like, I need to know that definition of this word, all of that, and then that would really shape what people at the Allen enter. Because, yeah, what information do you need to understand from a novice perspective? Okay, we are at the hour. I'd like to discuss next week briefly, unless there are questions on this. Okay, so last week there was a very interesting event on AI at the Institute, and I connected with some very smart individuals that have been using AI tools to build analysis infrastructure and help scientists. And I invited them over to present next week. I think you're really going to enjoy this. The Allen Institute for Artificial Intelligence, AI2, is a sister organization to the Allen Institute. And they've been developing something called, for instance, Semantic Scholar, that allows you to browse the scientific literature and make connections between papers using AI tools. And the team put together something called ASTA, which I found really interesting, that is using LLM to help build analysis and run code and create reports along different dimensions of hypothesis about what's in the data. And they are really interested in collaborating with us to see how those tools could help the science move forward. And so I invited them over to present next week. So this is first name is Pavithra, and then this is Bodhisattva. That's right. So next week we'll have I propose we have a presentation from most folks, and then you can ask them questions. And what they want to do is they want to try out running their tools and make the output available to everyone here so that we sort of see if there's something interesting that help us do this analysis in any directions. So so this way we'll be like at the cutting edge of open science and artificial intelligence. Any questions on this?
One quick question. Are they only using LLMs or other types of AI agents?
I don't know the answer, but you might ask them.
I will
but I
save
think
my
it's-
question till next week, but I think LLMs have a limited horizon in applying these to analysis. They're just, I'd like to hear more. Yeah.
That's right. That's right. I mean, I'm sure there are limitations, and but I see this as an experiment in its own. Like, okay, what can I get out of this? Is it useful? I think they generate codes that they run and then provide report on it, and they help. They've built something to sort of try to sort of structure it in a little bit as a scientist who sort of evaluate output, and it's very systematic. So I think it might be interesting. It might be not. We'll see.
Yeah. Thanks for inviting them.
Yeah, but they're really excited, and I'm really curious to see how you can help this project. Any other questions? So we'll have this event next week, and perhaps in 2 weeks, once the data is more available, you have more questions on how to start the analysis, and so that will be maybe a more brainstorming session in 2 weeks where we go over each one's items and see if what you need to get started. Okay, thanks everyone. Sorry, this was a bit of a long monologue, but
Thanks a lot. Bye.
Thank you. Bye.
thank you.
Thank you. Bye bye.